It's the 22nd of October, 2021. This is a RheumNow Podcast. Hi, I'm Dr. Jack Cush, Executive Editor of RheumNow.com. You know, sometimes I get on here, it seems like he's just moaning and griping and he's got an issue. And, you know, today I just have a few questions. Pretty simple, straight up. Is Tramadol really a weak narcotic? Should we really still be doing placebo-controlled trials? And what are we going to do about our patients who are smokers? So let's start with a study of placebos and what's happened to placebos in patients taking placebos against an active drug for psoriatic arthritis. You know, a long time ago, maybe 15 years ago, when the ACR20 was applied to PSA trials, they had single-digit placebo response rates, like 6%, 5%, 7%. You know, and for me, that was validation that the ACR20 was a good measure for PSA, when in fact it was devised for RA. But you know what's happened over time? There's been some placebo response rate creep. No, they're not creeps. The rates have gone up over time. And so this particular study looked at a pooled analysis of 42 trials and showed that over time that there has been an increase in the placebo response rate. It's now, I think, at 20.3%. I think it's the pooled average right now. But in the last 5 years, it's gone up roughly about 15% a year. The interesting thing here, however, is it hasn't affected the drug effect or the treatment effect. Treatment effect is the response to the drug under study minus the placebo response. That's the treatment effect or drug effect. The treatment effect and drug effects have remained sort of constant. So that means that as the placebo response rate goes up, so does the response to the drug. Again, placebos are wonderful drugs if only we could harness how we could use those in our practice and tell patients you're getting a placebo and see if they get better. I found an interesting comparison of the drug safety comparing two narcotics. I use a lot of Tramadol. I use a little bit of Codeine. I don't use any hydrocodone anymore. Oddly, when I send patients who need stronger medicines to pain docs, they don't give them the strong medicines they need. They want to do procedures. It's kind of a mess, honestly. Pain management really is a bit of a mess. Congratulations to any of you who are taking this head on. But in this particular study, it's a safety comparison of a very large cohort. I want to say there's like hundreds of thousands of patients in this thing. And straight up, when they got, it was 115,000 match propensity scored cohorts, Tramadol and Codeine. And head-to-head, Tramadol had significantly higher risk of all-cause mortality, 2.31 hazard ratio. Wow. A higher risk of cardiovascular events, a 15% higher risk, a hazard ratio of 1.15, and a significantly higher risk of fractures, 1.50 hazard ratio. No differences in falls, delirium, constipation, opioid abuse, sleep disorders. But, you know, I always say that, you know, Tramadol is a weak narcotic. Don't worry about it. Well, this kind of data makes me worry about it. It makes me worry about whether it is safe to use this drug in patients who need it for pain. Again, if you had another option, this might not be a tough discussion. But since we don't have many options, it's a tough discussion. Medscape had a nice review of an article about lupus patients taking aspirin, and that aspirin significantly lowered the risk of preeclampsia, a major problem with lupus patients who become pregnant. Interestingly, not only did aspirin lower the risk of preeclampsia, so did hydroxychloroquine. The risk factors for getting preeclampsia in their study was high disease activity during the first trimester, odds ratio of 4.5, BMI greater than 30, odds ratio 6.1, antiphospholipid syndrome, odds ratio 8, and history of prior preeclampsia. Guess what? You're going to get it again. An odds ratio of about 10. So yes, your lupus patients probably should be on aspirin during their pregnancy. Not after week 35, right? That's when any nonsteroidal or aspirin could contribute to premature closure of the ductus arteriosus, cause fetal distress. You don't want to do that. The recovery study in the UK looked at a matched cohort comparison of colchicine patients. There's a lot of patients in the study. Again, big numbers. Colchicine versus placebo, both groups got usual care. We have reported previously that it looked like colchicine probably had some protective effect. This very large study shows no effect. No effect on 28-day mortality, 21%. No effect on mechanical ventilation or death, 25% in both groups. Time to discontinuation, a mean of 10 days in both groups. The number of patients who were discharged from the hospital at day 28 alive was 70% in both treatment groups. Doesn't look like colchicine really does have much an effect. So congratulations to the EULAR guidelines, who said there's not enough data or robust data to include it or to recommend it. I'm not sure if you're getting in these arguments. I'm getting a lot of arguments and discussions these days about, are we close to herd immunity? What is herd immunity? How many people do we have to get vaccinated? Can we be running around without masks? The numbers are hard to follow here. JAMA had an interesting report this week where they looked at like 1.5 million blood donor specimens from around the country, representing about 3/4 of the country. And they had a lot of statistics about seroprevalence and whatnot, but the combined infection seroconversion and vaccination seroconversion as of May 2021 was 83.3%. So we're obviously not in herd immunity, and we've got 83% of people somewhat protected. The problem with infection seroprevalence and seroconversion is it doesn't last, and that's why patients who get infected need to be revaccinated or vaccinated at least there's some argument about the 60 or 90 days after the infection resolves. But we're getting closer. We need another 30, 40 million people in the United States to be vaccinated. Early on, we were talking about herd immunity and what it was going to take, and no one really knew a number. Some were throwing out 70%. Some were saying 80%. Fauci and those people who are supposed to be the experts weren't being pinned down because they correctly pointed out that we don't know the number. The number's different for different bugs. And this bug has had different levels of virulence, now with the Delta variant being the most virulent. So it's as long as there's a reservoir for this bug to land, it's going to be hard to achieve herd immunity. So I'm guessing we got to be over 90% vaccinated and or infected to get there. So again, keep up the effort and get everybody vaccinated. And as we always said, mask up, back up, wash up. Head-to-head, which is a better drug in psoriatic arthritis? Ustekinumab or a TNF inhibitor? Used a lot of TNF inhibitors. I don't know if you've gone to ustekinumab, the IL-12/23 inhibitor. Very effective, in my opinion. But head-to-head, real-world study, almost 900 patients. If you looked at minimal disease activity, it was 26% with ustekinumab, 31% with TNF inhibitors. If you looked at CDAPSA, Joseph Smolin, popular psoriatic arthritis, skin and joint outcome deal, 15% with ustekinumab, 19% with TNF inhibitors. The edge goes to TNF inhibitors, but ustekinumab not far behind. Again, those were not statistically different, numerically different maybe. The FDA approved this week another adalimumab biosimilar. Actually, they've already approved this biosimilar, Siltezo. This comes from Boehringer Ingelheim. It was actually approved 2 years ago, I think, but it's now the first biosimilar, TNF inhibitor biosimilar, that is interchangeable. And you may have followed this a few years ago. We were talking about interchangeability. They're working on a guidance document, and what this means now, the drug's already been approved as biosimilar. Now it's approved as to be interchangeable with the original compound, Humira adalimumab, and that means that the pharmacy or your hospital system can make the substitution without notifying you, and that it has met that criteria. We don't have a lot of biosimilar use in the United States, but this is, I think, gets under the doctor's skin quite a bit when your ability to control prescriptions is taken away from you, and the decision is made by money. Again, I do believe biosimilars are effective. I'm not wild about this interchangeable rule. Smoking Impairs Methotrexate Efficacy. We reported that this week. I don't know if that surprises you. We know smoking's bad for RA. We know smoking's bad for everything. Last week we said smoking's bad for Still's disease. It's bad for lupus, JIA, spondylitis, etc. But in this particular study, these are the results of the CAMERA-2 study. It was an early RA. CAMERA was a computer-assisted treat-to-target study, patients with early RA starting on either methotrexate with or without 10 milligrams of prednisone. And the treat-to-target part was escalation of methotrexate dose. When they looked at patients in the study who were treated with methotrexate, either with or without prednisone, what they found that being a current smoker was associated with a higher -28 over time, and that prednisone didn't factor into this in any way. Overall, current smoking lowered your risk of lowered your risk of having disease control by 43%. So again, not very good. What are we doing about this? I mean, I'm not sure what you do. When I see a patient who smells of tobacco, who's got cigarette stains on their fingers from tobacco, who freely admits to tobacco, they usually get like the 4-minute lecture about this is like gasoline on the fire, and this is something that they can control. And all the drugs in the world may not be enough to control their disease. But maybe we need to go further. Some rheumatologists have smoking cessation clinics within their own operation. Maybe you should double down on this whole issue and say, well, I'm really worried about your lungs and your heart. I'm getting a chest x-ray. I'm getting a calcium score by chest CT. I'm getting PFTs. I'm sending you to the pulmonary doctor. If you want to continue to be a smoker, you got to hear it from the people who are also going to tell you about how this is slowly killing you. I'm going to tell you how RA is going to slowly kill you because you're not stopping the thing that you should stop. And again, I have to recognize that it is incredibly difficult to stop. The basic rules here are that success rates are highest when patients join a smoking cessation group like Smoke Enders, use medicinal help, drugs that can help them beat the habit in the first 12 weeks, and probably getting the support and encouragement of their doctors. Again, I think this is going to be your responsibility, and you can engage these other doctors, but this is something where you can really make a big difference on the outcome. Our best drug, our anchor drug, is being handcuffed by the patient's habits. So a big study was published at the end of last week. That's the TICOSPA. That's a tight control in spondyloarthritis trial. This is 160 patients with axial disease, axial spondyloarthritis, relatively early disease, less than 4 years. They were either randomized to receive usual care by the rheumatologist every 3 months or monthly assessments and a treat-to-target regimen. And the outcome here was 1 year, 48 weeks. Oddly, they chose to have a functional outcome, and that's kind of the big deal in this particular study. Other treat-to-target studies that you're aware of, TICORA and several others, use a disease activity measure as the goal. Achieving the goal was usually a -28. In this study, they didn't choose a goal like an ASAS 40, ASAS 20 response or a BASDI response. They chose the consequences of that disease activity. They chose the ASAS health index, which is a functional outcome. So it's the ASAS high being greater than 30%. And guess what? At the end of 48 weeks, the treat-to-target group was numerically better, 47% versus the usual care, 36%, but that was not significant. So they missed. So treat-to-target didn't work. Well, it looked like it might have worked. Wait a second. It did work if you look at the other outcomes, the activity outcomes. ASAS 40, 52% versus 35%, highly significant. Other things that were highly significant was ASAS LDA, ASAS 20, BASDI 50. There were some that were not significant. That include MD Global CRP, BASFI, and other scores I know nothing about, but they're in there anyway. So does tight control, is tight control the goal in axial spondyloarthritis? Heck yes, and it does work. Depends on how you measure it. We're going to end with a study of a CD20 targeted therapy, Obinutuzumab, in proliferative lupus nephritis. As you know, there have been other B-cell targeted therapies that have failed in lupus, often without lupus in general and not lupus nephritis. In this particular study, 125 patients with lupus nephritis, that was mostly class 3 and 4 disease. Some might have had class 5, but it's mostly class 3 and 4 disease proven by biopsy. These patients were on a background of mycophenolate and steroids, and they're randomized to receive either placebo or Obinutuzumab on day 1, 2, 24, and 26, and they're followed for 2 years. The primary endpoint was the complete renal response at week 52. At week 52, there was a numeric improvement on Obinutuzumab, 35 versus 23%, not significant. However, if you followed them out to 104, it was significant at 41% versus 23%. So, it also improved proteinuria and GFR numbers and serologies with lowering of double-stranded DNA and C3 and C4 levels normalized. So it looks like this might be the next big thing in lupus. We'll probably see more studies on this at the upcoming meetings. That's it for this week on the podcast. I hope you enjoyed this. Go to the website to check out these citations and more. Be sure to follow us during ACR week. We've got a tremendous amount of work being done to give you the best coverage possible. You'll be hearing more about that in the weeks to come. Listen up.